Many of us watch our dogs as they progress from puppyhood to their senior years. But what if their journey through life holds significant clues about our own aging process? A recent study suggests that it does, and here we're going to examine how.
It's a compelling question. Researchers are increasingly turning their attention to companion dogs, not just as pets, but as a unique biological model that could help us unlock some of the persistent mysteries of Geroscience, that is, the scientific study of aging. To appreciate why, it's first necessary to understand a fundamental challenge that exists within aging research itself. This comparison illustrates the core issue. On one side, we have traditional laboratory research, which often relies on lab animals in highly controlled, sterile settings with limited genetic diversity. On the other side, we have real-world aging, the way we and our dogs experience it. Our aging process is shaped by a complex interplay of genetics, diet, and environment that laboratory settings cannot fully replicate. This creates a gap between observation in the lab and what occurs in reality. And it's precisely this gap that brings our canine companions into the scientific picture. The reason dogs are such a valuable model is that they serve as a near-perfect parallel for human aging. Think about it. They share our homes and are exposed to the same environmental factors. They often receive sophisticated veterinary health care. They also possess an incredible range of genetic variation across different breeds. And, very importantly, their shorter lifespans allow researchers to observe the entire aging process on a compressed timeline. To leverage this unique opportunity, a large-scale initiative called the Dog Aging Project was launched. This is an open science study monitoring tens of thousands of companion dogs across the United States. For this specific analysis, the scientists concentrated on a detailed subgroup known as the precision cohort, examining blood samples from 784 dogs. The composition of this cohort illustrates the study's strength. This was not a small, uniform group. The data represents dogs from 49 states, covering 110 different breeds, with an age range from less than a year to 18 years old. This level of diversity is critical for identifying aging signals that are universal, rather than specific to a single breed or location. So, what did the analysis of the blood from these dogs reveal? The researchers began looking for specific chemical signatures that were tied to the aging process. To find these signatures, they studied what's known as the metabolome. You can think of the metabolome as the complete set of small molecule chemical fingerprints left behind by all the metabolic processes in the body. It provides a real-time snapshot of an organism's physiological state. The initial findings were quite significant. The data showed that approximately 36%, so more than a third of all the metabolites measured in the dog's blood, were significantly associated with age. This finding confirmed that the metabolome does, in fact, change predictably as dogs grow older. The investigation became more focused. Within that 36%, one particular group of molecules emerged as a potentially new and potent biomarker for aging. These molecules are called post-translationally modified amino acids, or ptmAAs. What is critical to understand is their origin. They are only created when the body breaks down pre-existing proteins. They are not synthesized from scratch. This makes them a direct signal of protein catabolism, or turnover, in the body. This raises the question, why would these byproducts of protein breakdown accumulate in older dogs? The next phase of the study draws a connection between these molecules and a key organ system. The investigation pointed directly to the kidneys. One of the primary functions of the kidneys is to filter metabolic waste products, just like these ptmAAs, from the bloodstream. The researchers hypothesized that age-related changes in kidney function could be responsible for their accumulation. The data supported this hypothesis. The ptmAA levels were compared to two standard clinical markers of kidney health, creatinine and blood urea nitrogen, or BUN. The results were clear. As the levels of these waste products increased, which indicates a decline in kidney function, so did the levels of ptmAAs. This showed a distinct positive correlation. In fact, the strength of this connection was notable. For some of the ptmAAs, the researchers calculated that changes in kidney function could account for up to 67% of the age-related increase. This points to a powerful physiological link. This brings us to the broader implications. What might these findings mean for the future of how we monitor health and the aging process? This quote from the research paper summarizes the findings concisely. The work has identified a robust new indicator of biological age in dogs and has also pinpointed a key physiological process, kidney function, that appears to drive this change. So let's distill the key takeaways. First, we have a new biomarker that may allow for more precise tracking of physiological aging in dogs through a blood test. Second, this provides a path forward for human health. A gradual decline in kidney function is a known hallmark of aging in most mammals, including us. While clinicians already test for markers like creatinine, these ptmAAs could potentially serve as more sensitive, earlier indicators of these age-related changes. It leaves us with a compelling thought. The study of our canine companions may not only improve veterinary medicine, but could also provide a unique window into our own biology, potentially guiding us toward a healthier, longer life.